Well, dear students, welcome to my next class. In today's topic, we will be discussing about plant breeding. I have given an introduction about this. In this class session, we will discuss about the general methodology. It can be asked like this also in your examination. The general methodology. Here we will find out the different steps by which we can create a hybrid plant. Now, first of all, I have told you the definition of hybridization also. What is hybridization? Crossing of two different parents. Two different parents means they are differing in their characters, in their traits, in their characteristic. Right? They differ from each other. For example, one variety will have disease resistant, the other variety will be having more productivity. Such parents are crossed to get a new generation in which both these characters should be there. Such a process is known as hybridization. So in plant breeding general methodology, that procedure we have discussed, it will be asked for 3 marks or for 5 marks also. In detail it is asked. Right? The first one is what is known as collection of germplasm. Collection of germplasm. What do you mean by this? Germplasm here refers to genetic variability. Genetic variability. What do you mean by that? For example, tomato plant will take. There are too many different breeds, different varieties of tomato plant. For example, what we say wild one, or in our language commonly we say nati tomato, right? Then hybrid one, transgenic one, round one, longer one, having thick pulp is a different breed, having thin pulp is a different breed, having different shapes, right? Size. So there are different breeds, different varieties. So we have to collect all those things. That's what is known as collection of germplasm. All the genetic varieties, the genetic varieties or variability has to be collected here before we go for selection process. We should collect all the variety, the present variety and the previous variety. Like what we call the natural one, most common natural one is what we call as wild variety. Present variety, hybrid ones. So all that genes has to be collected, their DNA has to be collected. Such a procedure is what is known as collection of germplasm. Germplasm, you all know germplasm, right? From which has DNA and RNA, from when the germination starts, the production starts. So all the varieties of that particular species, of that particular breed, whether it's a wild variety or the present variety, different one, shorter plant, longer plant, having the different types of fruits in that, should be collected. Such a procedure is known as collection of germplasm. After that, once you collect here many germplasm, many varieties you have collected. What you have collected? Many varieties of the same plant you have collected here. Then, after that, selection of parents. Selection of parents. Now, why we call them as parents? Because these two varieties, for example, one is a variety, the other is B variety. Now you have C, D, E, etc. You have so many varieties of same plant you have collected. Now, for example, this A variety is disease resistant. This B variety is high yielding. This variety is resistant to diseases caused by viruses. For example, viral diseases. This variety is resistant to that. It is not prone to, or it is not subjected to viral infections. Why? Because this variety is specified for viral infection. That's one characteristic we are looking for. Second characteristic, this variety gives what? High yield, more productivity it gives. Now what we have to do? Such parents among all these varieties we have collected in germplasm from many varieties, two different parents which are different in their characters should be selected. That's why we call this as selection of parents. Selection of parents. Now, for example, we have selected one which is disease resistant, other as high yielding. Now, cross has to be done with this. Cross has to be done among all these. Right? Cross has to be done. Third procedure is hybridization. What is it known as? Hybridization. Now, what is hybridization? It may be asked for 1 mark also definition. The cross between two parents are here as far as this topic is concerned. The cross between two plants which are differing in their genetic characters. What is the character? One character disease resistant, other character high yield. This C variety may be dwarf variety. This D variety may be tall variety. This E maybe having early maturation. Different characteristics are there. Among them, we have selected two plants. One is a disease-resistant plant. The other parent is high yielding. Now the cross has been done. So what is hybridization? Hybridization, the process of crossing two parents differing in their characters, to get the progeny, the next generation. What I studied in genetics, F1 generation, to get the next generation which has both these characteristics. This character also should be there. This character also should be there. Both should be there. Such a process, such a procedure, such a method is what is known as hybridization. Now here, after selecting, what we are going to do? We are crossing them. What we are crossing? Crossing them. Hence it is known as cross hybridization. Cross hybridization. So we are crossing between the two parents, same breed, same breeds, different species, different varieties. So this is variety. This is written. For example, this is also tomato. This is also tomato. All tomato. For example, I am taking because we are the second largest. We have the second largest market in the whole Asia, Kola, where we export tomato a lot. Many people, many students know about this. Classical example. For example, this tomato variety is disease resistant. For example, like take the example of leaf curl virus. That is resistant. Now this is high yielding. It gives more productivity per plant. Many number of fruits are being bear. Tomato is also a fruit. Whatever that comes out of flower is a fruit. So this variety is selected. This variety is selected. Now you are crossing between these two. You are getting a variety known as AB, which is both what disease resistant also and what high yielding also. Both these characteristics you got in one. Such a process is known as cross hybridization. Now you get here F1 generation, or the generation you get here, right? Then the next generation you get here. Then after that, testing and selection. What you have to do here? Back cross. What has to be done here? Back cross. All this terminology you have studied in genetics chapter. Back cross, F1 generation, filial 1 generation, test cross, back cross, self pollination you have studied in first chapter. By masking, by emasculation, by bagging, by self pollination. Right? By self-pollination, all this terminology, you know that, very basic ones. By self-pollination, right? This has to be, cross has to be done. By self-pollination among this, right? Yeah, this is just to understand, back cross, self-cross, F1 generation, all those things. By using self-pollination, you have to cross them, you have to produce many of the same breeds, many number of copies of many plants. By using self-pollination, these plants are being made. That's what is known as, right? That's what is known as testing. After that, we have to test, right? Testing and selection of the breeds, testing and selection of the progeny, This is the third step, followed by the fourth step, what is known as testing. Why testing? After you get this, whether this plant, what we have crossed, what we have hybridized, bearing, having, possessing both the character. What character? One we are looking for disease resistant, other we are looking for high yielding. These both characters should be there. Such plant we have to test. How to test? By growing in the fields. In the lab condition, this will show. But whereas from lab to land condition. Why I am telling these two words now here? Because in the next topic, in plant tissue culture, this you have to understand. So here in the laboratory, when hybrid plant is made, when transgenic plant is made, result comes that it shows both the characters, but you will come to know the result only when you plant these plants. When you plant these plantlets, right? What you get in nursery, those plantlets you have to test in different fields, having different seasons for at least for 3 years or 3 seasons you have to test them. Then when after testing for many times, when they repeatedly show disease resistant character is also existing, high yielding character is also existing, fine, clear, both these characters are expressed in a single plant, then it is a successful breed, successful cross, successful hybridization. Then such a plant, what is the fifth step, then it will be released in the market, commercialization, after testing in the fields, many seasons, in different fields, for example like Kolar district, Tumkur district, Bangalore district, Chikmagalur district, then in different states, different seasonal conditions, different temperature conditions. What is our this one here? This is resistant. Whether it is resistant to diseases or not, fungal infections or not, or viral infections or not, or bacterial infection, whichever the characteristic. Okay, in one season it has given more production. whether it's giving the second season also it is giving more production or not. This has to be tested. Why? To understand whether the genetic expression is occurring or not. These two characteristics which has come in one plant, whether these two characteristics are expressed or not. Genotypically they will be present, but whether phenotypically they are expressed or not. Whether this plant is disease resistant when it is grown in different seasons, different fields, whether it is giving high yield even after 2 generations, 3 generations. Many times you are planting these trees in many different fields. You are planting that field is known as cultivar. What these farmers will call cultivar. Before cultivation, before giving it to market for releasing, it has to be tested in different fields. After testing, once it is very much confirmed that these plants are giving good result, both characters are expressed in a single plant, such plants will become a simple name. Like for example in case of tomato you have many namdari, you have different one, like for example papaya, dwarf variety, it is known as red lady. Very simple name, very catchy name they used to keep, so that mouthword marketing should occur. One farmer should tell the other farmer, 619, one variety of the tomato. Like this, very simple names they'll keep, so that it should be catchy also. The farmers should remember that, because one farmer will blindly believe the other farmer. If you a scientist or a person goes and tries to make them understand, they will not trust you. Whereas a farmer says, I have used this product gives good yield, fine. Another farmer blindly goes, take that, fine. So mouthword marketing should be there. So before releasing into the market, commercialization, before it is commercialized, that has to be tested in different conditions. Once it is tested, confirm both the characteristics are being expressed in many seasons, different seasons, many a times, without inbreeding depression, then such a product can be named, simple name can be kept to that variety. Like for example, I told you, for papaya variety they are kept as red lady. Very easy, very simple, everybody remembers it. Fine. One time I told you, you also remember it. So such a short plant of papaya, having very good fruits, small fruits, red in color, such a variety will be kept. One catchy name will be kept, easy simple name will be kept, and then it will be commercialized, marketed, so that it should reach the normal farmers, right? So here in this topic we have covered the general procedure of hybridization. That is nothing but plant breeding or hybridization, collection of germplasm, where we are collecting many different varieties of the same species, different varieties of the same breed. Then after that, among many characteristics, you select the two characteristics, that is the selection of parents, and after that you go for hybridization. After hybridization, you go for testing whether this what cross we have done, what character we have done, whether it is being present, expressed or not, testing, and then selection of the progeny. After that, it is being released and marketed. Very simple procedure, very easy words are there, simple terminology, simple procedure. This is the basis for your transgenic plants in the next topics. So hybridization technique is a general technique in the plants. This is what I have discussed. Go through with our study material. Keep studying. God bless you. Good luck. Take care.